Welcome to Introduction to Biology. This is Dr. Urbi.Ghosh. In this lesson, we will discuss Mendel's experiments using purple and white flowers which he crossed. These experiments laid the foundation for modern-day genetics and for understanding dominant and recessive traits, which are part of our genes and way of inheriting things. When Mendel crossed a purple flower, for example, with a white flowered plant, he found amazing things from the first generation, which was that they were all purple. So when he took a true breeding purple plant and he crossed it with a white breeding plant, he found that in the first generation was all purple. Now, why could this possibly be that everything is purple in the first generation and there's an absence of white? The reason was that all the plants in the F1 generation, or the first filial generation, were purple was because the purple color trait was dominant over the white color trait in these flowers. And so then in the F2 generation, or the second generation of filial generation, there was white flowers again. This meant that the color purple was masking over the white color. We now know this is due to the passing of genes both in recessive and dominant form. So at the time, Mendel called these genes factors, the thing that carried traits from one generation to the other. So what about the disappearance of the white flower trait? In the F1 generation, the trait of the white color flowers were not seen at all. This was explained by Mendel in a very elegant manner. He thought that these factors, in the presence of another factor, which we know are genes or alleles now, that some genes are always kind of expressed irrespective of where the other one is there or not. And these genes are called dominant genes. The others which are suppressed are referred to as recessive genes. So why did recessive genes not express themselves in the F1 generation? Well, let's consider the fact that all these purple flowers had the big P, or purple, dominating over the white. And it was only when you crossed a little p with a little p that you got the true white flower. So let's take a closer look. What he did was he took the purple flower, which is the homozygote for the purple plant, and he crossed it with a little p, which is the white homozygote. And so what we have here is in the first generation, everything is purple. But what you get is a big P, which comes from this parent, and a little p, which comes from this parent. So they have the little p in them. It's just that they're not being expressed because it's being dominated by this big P. In the second generation, when you cross the big P, little p with another big P, little p heterozygote, and they're called hetero, meaning two different genes, whereas homo, meaning two similar genes, you get a big P, because this one gave a big P here, and a big P came over here, and this one gave a big P here, and this one gave its little p over here. When you cross these combinations, and this P came from this one, and this one came from this one, you find that the appearance of the white color comes out because this particular plant does not have a big P in it. So the recessive trait appeared to have passed through the F1 generation monohybrids unchanged and into the second F2 generation. And this happened trait after trait, whether it be the color of the flower, the seed shape of the pea plants he studied, height, whether it be tall or it be short, whether it be seed color, whether it be yellow or green, or a pod color versus yellow and green. So he was able to find this happening consistently over generations and over different traits. So the F1 generation, what happened is a purple is crossed with a white, and you get all purple flowers. And in the second generation, you have the purple flowers being crossed, and you get some purple and white, and that's because the blending of inheritance was not a variable explanation, according to Mendel. Inheritance appeared to be particulate in nature, and some versions of the trait mask other versions of the trait. And this he considered to be a dominant trait. And the traits that were masked in the first generation were considered recessive. And he found that the F2 ratios were consistent trait after trait after trait to be 3:1 of dominant being 3 to recessive being 1. And this suggested that the F1 monohybrids must have carried the trait in some particular way, but it was hidden. And the ratios further suggested that heredity particles segregated into gametes and they came back together in fertilization.